The purpose of mitosis is to maintain the correct chromosome number for the species and for an individual. Eukaryotic chromosomes are made up of chromatin, and chromatin is just like this unraveled chromosome that is a combination of DNA and proteins. They're all spread out in the nucleus of a non-dividing cell so that transcription can occur and then translation. They condense into compact form for cell division. Each species has a characteristic number of chromosomes. We refer to the diploid number or the 2N number as the cells who have 2 pair of each type of chromosome. And in our body cells, we have 46, which are 23 pair. Our haploid number is the 1N, so half of the diploid is the haploid. Cells have half the diploid number of chromosomes, and we have these in our sperm and egg cells. So we have 46 total chromosomes, 23 pair in our diploid cells, which is every cell in our body except for egg and sperm. And we have in the egg and sperm, we have 1N, or half that number, which is 23 chromosomes, one member of each pair. Let's look at an overview of mitosis. Nuclear division in which chromosome numbers stay constant. So one 2N cell becomes two 2N cells. The DNA is going to replicate and duplicate. Each duplicated chromosome is composed of 2 sister chromatids, and they're held together by a centromere. And we'll look at a picture of that in just a minute. Sister chromatids are genetically identical, and during mitosis, the centromere divides, and each chromatid becomes a daughter chromosome. So here we have a chromosome that's made of 1 chromatid. Replication happens, and you have 2 sister chromatids. They're genetically identical, and they're held together in this region by the centromere. And during cell division, these sister chromatids are going to split apart, and they'll be seen as chromosomes with 1 chromatid. So here's the centrioles, here's the chromosomes within the nucleus, and DNA replication happens in the nucleus during interphase, the S phase of interphase. And you end up with duplicated chromosomes that are made up of sister chromatids. So we have a 2N equals 4 and a 2N equals 4. They're just duplicated here. And in mitosis occurs, and you have the same thing. You have 2N equals 4, 2N equals 4. So we have replicated, and then we have divided, and still end up with 2N cells, which is very important when it comes to mitosis. The phases of mitosis have been split up into 4 general phases, prophase being the first, and it's important that you know what's going on, the general idea of what's going on in each phase, and why it's going on. So prophase is the nuclear membrane is going to go away, it's going to disappear, the centrosomes are going to migrate, the spindle fibers are going to show up, the chromatin is going to condense, and the chromosomes now look like chromosomes and not just like DNA strung out. And you have to remember this is after the stage of interphase, the synthesis stage of interphase, where we have replication of DNA. And each of the chromatin that's condensed into chromosomes is made up of 2 sister chromatids. The centromeres are going to attach to the spindle fiber. So all this happens during prophase. Now, even though we're going to divide mitosis into 4 general phases, what you have to remember is that each phase, it's a constantly moving cycle. So we just get glimpses or pictures of each phase. So this picture shows us what early prophase would look like, what prophase, middle of prophase, and late prophase would look like as it's gearing up to go into metaphase. And this is after it's come from interphase, from the G2 phase of interphase. And these are actual pictures here of mitosis, cells in mitosis in early prophase, prophase, and late prophase. We look down here at the pictures that they've drawn, and you can kind of see where the similarities are, so it's easier for you to view in the actual cell. So we have the DNA in the nucleus right here, and the asters showing up here and here, which are coming off the centrosomes. And the nuclear membrane starts to break down, so there's more room for this to happen inside the cell. We start to see the spindle fibers start to form a little bit more, so that's what's going on here. Here you see the where the asters are on either side. They've migrated on either side of the cell. We see the duplicated chromosomes within the cytoplasm, which we have right here. And in this late prophase, beginning of metaphase, we start to see the chromosomes line up and get ready to go through actual division. Look at metaphase. Metaphase is easy to recognize because the chromosomes line up at the metaphase plate, or at the equator, as it's said sometimes, and it's very recognizable. In anaphase, the centromeres divide and they start to pull apart the sister chromatids to opposite poles, and it's easily recognizable as well. Telophase, the nuclear membrane starts to reform, the spindle fibers disappear, and cytokinesis occurs, which is the cell cutting. And actually, telophase is a reversal of prophase. So we start to see the nuclear membrane start to come back, the chromosomes start to not be as condensed, and we start to have two new functioning cells that are separate from each other. And that's what this picture shows. As I said before, metaphase is very recognizable whether you're looking at a drawing or whether you're looking under a microscope. And you notice how those chromosomes line up in the middle in metaphase. They pull apart in anaphase. And sometimes it's difficult to tell anaphase, late anaphase, and telophase apart. But you notice here, this is telophase because we start to see cytokinesis occurring. There's a cleavage furrow that happens in animal cells and allows for those cells to split apart. You see the nucleuses reforming here, and you can't see that so much here, but that's what's going on. So we're starting to see a reversal of prophase. The spindle fibers have gone back into the centrosomes, and we're starting to have full functioning two new cells in interphase again, ready for whatever their function is going to be. Mitosis in plant cells and animal cells is a little different. Still the same basic phases occur, but they are just a little bit different. They occur in what's called meristematic tissue, and plants don't have centrioles or asters. So we have chromosomes here in prophase, and you can see that they're double-stranded. You see the cell wall, and I know it's not as clear, but there's the cell wall outside of it. Here's metaphase, and we have spindle fibers that are showing, and we have the cell wall again, so they're lined up in the middle in metaphase. Anaphase, you can tell that they're pulling apart. And in telophase, there's a cell plate. Instead of the cleavage furrow that we saw, there's a cell plate that's laid down, and you start to see the reversal of prophase again, where you start to see two different nucleuses form. Cytokinesis in animal cells, you start with the cleavage furrow between the daughter nuclei, and you have a contractile ring that deepens the furrow, and then this is going to happen until you have two complete cells. In plant cells, there's a new cell wall between daughter cells. It's a flattened area with small discs that appear between the daughter cells, and that forms the cell plate. Then vesicles release molecules to build the new cell walls. And the vesicle membranes complete the plasma membrane, so it's a little bit more complicated in plant cells than it is in animal cells. That concludes mitosis. Remember that mitosis is happening in every one of your body cells that actually go through the process of replication. Not all do, but it maintains the chromosome number, which is 46 or 23 pair for humans, and happens in every cell in your body except for your sex cells, which will be covered in meiosis.